Hey everyone, so this video is going to be the second half of the metabolism video, and it contains information on cellular respiration. By the way, if you haven't seen the first video and you're interested in seeing it, I'll have that link down below in the description box. It contains information on glycolysis, fermentation, and a little bit of photosynthesis. Anyway, let's get started with this one. Now let's look at cellular respiration. With cellular respiration, there are three stages, which include the transition step, which is also called the intermediate stage, the Krebs cycle, which is also called the citric acid cycle, and finally the electron transport chain. And before I get into the details of each stage, some things to keep in mind are not all organisms can undergo cellular respiration. Also, both the intermediate stage and the Krebs cycle take place in the mitochondrial matrix for eukaryotes, and for prokaryotes, it is the cytosol or cytoplasm because they do not have mitochondria. The electron transport chain, on the other hand, occurs in the mitochondrial membrane for eukaryotes, and for prokaryotes, it is the cytoplasmic membrane because, again, they do not have a mitochondrion. And lastly, cellular respiration is an aerobic process, meaning it requires oxygen for it to happen. In the case where an organism can perform cellular respiration, instead of undergoing fermentation to regenerate NAD+ and continue the cycle of glycolysis, chances are the organism is going to go ahead and perform cellular respiration instead, because as I mentioned before, that is more efficient. That said, remember that one of the products of glycolysis is pyruvate, and that it is an energy-rich molecule which can be used in the generation of more energy during cellular respiration. However, pyruvate cannot be used directly in the Krebs cycle, which is where the intermediate step comes in. As the name states, this process is an intermediate, so it links the process of glycolysis to that of cellular respiration. And because pyruvate cannot be used directly in the Krebs cycle, the goal of the intermediate step is to convert the pyruvate into acetyl-CoA, which can be used in the Krebs cycle. So how does this process work? Well, pyruvate loses a molecule of CO2 and NAD+ carries electrons from pyruvate, and thus it is reduced to NADH. In addition, a compound called coenzyme A will be added to the pyruvate, which produces acetyl-CoA. So now I'm going to talk about the products of the transition step, which are 2 molecules of acetyl-CoA, 2 molecules of carbon dioxide, 2 molecules of NADH. Also remember that there are 2 pyruvate molecules produced at the end of glycolysis, so each of them will be converted to the products of acetyl-CoA, carbon dioxide, and NADH, hence why there are 2 of each of the products. Moving on to the Krebs cycle, which is the second stage of cellular respiration. The goal of this stage is to extract the energy present in the bonds of acetyl-CoA. This energy is termed as future energy, as it will be used later on in the electron transport chain. And the energy extracted is in the form of the electron carriers, NADH and FADH2. That said, immediate energy in the form of ATP is also produced, but it is very small. Also remember that from the intermediate stage, 2 acetyl-CoA molecules are produced, thus each of them will be used in the Krebs cycle, and so the Krebs cycle is a process that occurs twice. And the products are multiplied by 2. So now I'm going to go ahead and tell you what the products of the Krebs cycle are. They are 2 molecules of CO2, 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of ATP, and 1 molecule of oxaloacetate. And when you multiply all that by 2, you're going to get 4 molecules of CO2, 6 molecules of NADH, 2 molecules of FADH2, 2 molecules of ATP, and 2 molecules of oxaloacetate. And again, you're multiplying the products by 2 because you're going to have to repeat the Krebs cycle twice. And by the way, oxaloacetate is what acetyl-CoA is converted to at the end of the Krebs cycle. And FAD is just another electron carrier like NAD+, which arises in the Krebs cycle. It will gain electrons and then be reduced to FADH2, just like NAD+ will gain electrons and then be reduced to NADH. And lastly, I mentioned that no ATP is used in the Krebs cycle or the intermediate step, unlike in glycolysis where you use ATP. Now we'll look at electron transport chain, which involves a series of electron carriers that transfer electrons amongst themselves until the electron reaches the final electron acceptor, which is oxygen. And oxygen is a final electron acceptor because it is highly electronegative, whereby it has a great affinity for electrons, meaning it likes electrons, so it pulls electrons towards itself. And if you recall, oxygen is one of the requirements of cellular respiration, and this is where it comes in. Also, remember that future energy I was talking about? That is energy in the form of NADH and FADH2. This is when they are used to produce more energy in the form of ATP. The energy that is released from the electron transfer amongst these electron carriers is used to establish a proton gradient or protomotive force across the mitochondrial membrane, which is what drives the production of ATP. And by the way, the proton gradient is simply a gradient of hydrogen ions. So I'm just going to explain how the electron transport chain works a bit further. And real quick, I just want to show you all a couple of structures on here. So this right here is ATP synthase, and this area here is the mitochondrial matrix, and this structure is the inner mitochondrial membrane, and this area here is going to be the intermembrane space. Also, this is the hydrogen ion molecule. That said, the electron transport chain is made up of electron carriers, protein complexes, and ion pumps. As electrons are transferred amongst the electron carriers, which are caused in the order of NADH to FADH to oxygen, the ion pump will pump the hydrogen ion out of the mitochondrial matrix across the inner membrane to the intermembrane space of the mitochondria. This is what creates an electrochemical gradient where there is more hydrogen ion in the intermembrane space. As you can see right here, you have more hydrogen ion compared to in the mitochondrial matrix. So the hydrogen ion will then move down the concentration gradient. That is, it's going to move from the area of higher concentration to the area of lower concentration. So it's going to move from the intermembrane space to the mitochondrial matrix via this enzyme, which I said is ATP synthase. And this movement is what is going to provide energy for the production of ATP. And the entire process is known as oxidative phosphorylation. Moving on, let's talk about the products of the electron transport chain, which are 6 molecules of water and between 32 to 36 molecules of ATP. And I say between those numbers because various sources will tell you either one of those numbers, and some sources do tell you it's 34. Anyway, I believe the one most commonly used is 32. That said, looking at this, you can see how much more efficient this process is compared to glycolysis. There you get a net total of only 2 ATP molecules, whereas here you get 34. Also, when you look at the reaction equation for cellular respiration, now that we've covered all of the stages of this process, you can identify where the various products come from. If you recall from the transition step, you get 2 molecules of CO2, and from the Krebs cycle, you get 4. And so when you add that together, it gives you 6, which is what you can see here on the right-hand side of the equation where the products are. Also, I just said the 6 molecules of water are gotten from the electron transport chain. And lastly, you get ATP from both the electron transport chain and from the Krebs cycle, although that of the electron transport chain is much more. Finally, to conclude this video, I'll leave you with a summary slide which basically shows the process of glucose catabolism from glycolysis all the way to the electron transport chain. It also shows the specific end products of each step, which will be used in the next step until you arrive at the desired products of ATP. Feel free to pause the video and take a screenshot if you need it. So that's it for this video. As usual, if you have any questions, leave them in the comments below and I'll get back to you. Thanks for watching, and I wish you the best of luck with your studies and any exams. Oh, and don't forget to like and subscribe if you want, and share the video with someone who you think might find it helpful. Bye.